What if I told you that the DNA inside you isn't really your own, at least not all of it? Around 8% of all our genetic information is from viruses infecting us and leaving their genome inside ours. And the really weird thing is that you probably wouldn't be alive without one of these ancient viruses inside you. In fact, humanity likely wouldn't exist at all without an infection tens of millions of years ago.
There were some hopes because certain scientific studies, we can say it's a study and also more of a kind of a scientific theory which talks about an arc gene or genome which could have played a profound influence on the growth of our brain and the development of who we are today many million years ago was largely instigated by a virus.
These viral fossils are buried in our DNA, but parts of these viruses are still very functional and in use. Our cells have taken some of the genes from the viruses infecting us and turned them into our own tool.
But looking at the human behavior with partial relaxation, this virus doesn't seem to have worked like that. With a burst of new intelligence, it's not happened. Hopefully, the virus will mutate itself into that kind of a virus that a higher level of brain power happens. Well, it is also noted that in the development of mammals, who are right now categorized as the highest level of evolution on the planet, development and evolution of ability to produce placenta is believed that it's the virus some 100 million years ago which could have instigated this process within very primitive mammals which existed at that time. This is the basis of nutrients passing from mother to child and hence a full-grown child being… no, not full-grown in that sense, but a full-formed child being delivered, whether it is a human being or a cow or a dog or all these mammals, has become possible mainly because of development of placenta, and that could have been instigated or has been instigated by some virus 100 million years ago.
Here is the story of how virus infections very likely led to the evolution of the mammalian placenta and gave rise to all placental mammals from cows, cats and elephants to primates and us humans.
So you don't know what treasures this virus is holding for you. Maybe your brain will develop or something else will happen, maybe you'll grow a horn. Because people are killing rhinoceros for a single horn, not even two. So if you grow your own horn, maybe the rhinoceros will have a party. In some way, we don't know what advantages will come out of it.
The placenta is an organ connecting the developing fetus and the mother inside the womb. The fetus's blood flows through the umbilical cord to the placenta. Viruses are tiny, infectious things. They have genes but no cellular structure, and they infect cells to make more copies of themselves. A virus turns a cell into a virus factory. Sometimes retroviruses have ended up infecting our germline cells and have been permanently made part of our genome. These integrated viruses pass on from generation to generation. This is why our genome has the remains of ancient viral infections. And now we get to the really interesting part. With viral genes available in us, our cells have made use of one viral gene in a really revolutionary way. The envelope glycoprotein coded by the ENV gene gives the retroviruses a way to fuse their outer membrane with the membrane of the cell they are infecting. The envelope glycoprotein, or ENV protein, recognizes a cellular membrane protein, anchors itself to the protein, and slips inside the cell. Cells gain the ability to fuse together. They can fuse together to make a sheet of cells that have multiple nucleuses and share all cell organelles. This sheet of cells opens up completely new possibilities for embryonic development, allowing for the formation of the placenta. More and more evidence supports that adopting the NF gene turned our ancestors from egg-laying mammals to placental mammals, drastically changing the way the embryo develops. With the evolution of the placenta, an embryo does not need to be in an egg, facing the outside world, but stays safe inside the mother, where only a thin lining of tissue separates the fetus's blood from the mother's blood. Outermost on this very thin tissue layer is the syncytiotropoblast, a layer of cells fused together via syncytin-2 protein. Syncytin-2 is the name we have given to the domesticated NF gene in our genome. The fused together syncytiotropoblast cell sheet is invasive, and is what implants the embryo to the wall of the mother's womb. Later, the syncytiotropoblast facilitates the flow of nutrients and oxygen between the mother and the fetus. And its functions don't stop there. It also produces hormones, regulates the immune response, and protects the fetus from pathogens. We probably wouldn't be here if it wasn't for a rare event around 40 million years ago. A retrovirus infected our ancient ancestor, and its genome happened to end up in the germline cells and spread through the population. It inserted itself in chromosome 6, giving us syncytin-2. This here is what part of the human syncytin-2 protein looks like, using X-ray crystallography to determine its 3D shape. This part here is key in the fusion of membranes. Syncytin-2 is not the only viral NF gene that we have turned to our use. In a separate rare event around 25 million years ago, another retroviral infection gave us syncytin-1, another viral NF gene that became part of us. More and more evidence supports the key role of virally derived syncytins in placental formation. The cells forming the syncytiotropoblast can't fuse when syncytins are blocked, and removing mouse syncytins completely disrupts the formation of their placenta. We humans have two virally derived syncytins, syncytin-1 and syncytin-2. Placental mammals are many, but very interestingly, most placental mammals don't share our syncytins. Instead, the incorporation of retroviral NF gene for mammalian placental formation has happened over and over. At least 10 times are known so far. So far, a syncytin gene has been found in every mammal studied, with different syncytins fusing placental cells in different mammalian groups. A model proposed by Thierry Heidmann and colleagues from the Gustave Roussy Institute explains the findings like this. The first syncytin gene domesticated came from a retrovirus infection around 150 million years ago. This syncytin gave rise to all placental mammals. The first syncytin has since been replaced by newer syncytins, unique to each mammalian group. Each new syncytin has given the species an evolutionary advantage. With enough time, millions of years, more rare germline infections leading to virus gene domestication have taken place, giving mammals a variety of syncytin genes. Not only have retrovirus infections likely initiated and shaped the evolution of the placenta of mammals, they've done so in non-mammals as well. Recently, the Mabuya lizard, a lizard that has a placenta and gives live birth, was found to have a syncytin gene of its own. As with the syncytin genes of mammals, this syncytin was able to fuse cells grown in a cell culture and was expressed in the placenta. With the placenta evolving multiple times independently, it seems like a successful strategy for embryonic development. The retroviruses in our genome are not merely along for the ride. They have drastically shaped evolution and likely brought about our existence. We are not only descendants of earlier primates, we are descendants of retroviruses. Thank you for watching. Check out the other videos on this channel and leave a comment down below. I'll try to answer all your comments. If you haven't subscribed, do so.
See you again.